Good morning, everybody. Welcome to the 42nd annual meeting of the American Association of Endocrine Surgeons. It is my great honor and pleasure to be your president this year. It's been really a tough couple of years with COVID, so I know it's hard not to be chatting to people you haven't seen in a couple of years, probably. At least certainly that's been the case for me. So it's a pleasure to be back here in person and back in a meeting. So welcome everybody, and I hope we have a great time. I think the agenda for the meeting is fabulous. I do have a couple of things. We are encouraging mask use. Obviously, when you're eating, that's not going to be possible. But in the meeting, it's probably a good idea to wear masks. I would like to introduce our officers. Cord Sturgeon, the president-elect, Fiemo Nweriaku, our vice president, James Lee, our secretary, Tracy Wang, treasurer, and Kepal Patel, our recorder. I've had the really distinct honor to work with these folks over the last year, and some like James and Tracy over the last couple of years. So it's really been a great honor and pleasure, and have made my job actually very easy. Our past presidents, Alan Sipperstein, who's also I've worked with closely now for the last 3 years, Herb Chen, and Martha Zeiger, who are all present, and obviously welcome them. And then our counselors, Brunesta Lindemann, Peter Zako, Denise Carnero-Pla, Carrie Lubitz, John Liu, and Jennifer Rosen. So those have been our leadership over the last year, and it's been an outstanding group. I also have to thank the AMR team, which without really none of this would happen. Lauren Santangelo, our executive director, Emily Williams, the project coordinator, Delphine Tanner, the events manager, and Margaret Cloyd, the association administrator. So again, you've probably seen them or met them each, or at least one of them. So they've really been a great team to work with. We have 2 invited lectures this year, as always. Tom Giordano from the University of Michigan. Looking forward to hearing his lecture in just a short while. And then our very own Chris McHenry, who will give us a historical overview of the contributions of Cleveland to endocrine surgery. I have to thank our supporters, and please do visit the booths. You have to realize this is key to our financial viability as getting this meeting off the ground. And so in addition to which, with all the new technology that we're going to hear about this weekend, and have already heard about for some, now is never a better time to familiarize yourself with these folks. There were some special educational grants. The RFA course was supported by an educational grant by Tai Wung. The ultrasound course supported by a educational grant from Verisight. And those of you who were able to avail yourself of those recognize how critically important those are. And then Checkpoint Surgical this year in a new endeavor to have increased involvement of our colleagues in Central and South America, supported a travel grant, and so we thank Checkpoint very much for that. We do have to take this time to have a small moment of silence, a short moment of silence for two of our members who passed away over the past year, Walter Goldfarb and Ed Poloyan. There are tributes to them in the program booklet, which you can read and should read. They were both distinguished members of the society and instrumental in the development of many of our current members, and so we do need to recognize that. So if we can just take a moment of silence for their contributions and their passing. Okay, on a much more boisterous note, I'd like to actually have Dr. Alan Sipperstein come up here. As you know, he was the president for 2 years during the COVID pandemic. He was absolutely fabulous in negotiating the trials and tribulations of the pandemic as it related to our society. It was a distinct honor and pleasure for me to be the president-elect underneath Alan and see how he negotiated and handled things, which was exemplary. And he really deserves more than just a few moments here on the podium. So, Alan.
Great. Thank you so much. I want to welcome my AES family to my city of Cleveland. I am so glad to see everybody in person. And yes, Tom, it was a tumultuous couple of years. I want to really thank the council and AMR who, over the past couple of years, have done a huge amount of navigation to really make our society whole. I mean, realize the meeting in 2020 was canceled at the last minute. After a lot of back and forth shuffling, we realized that in 2021 we had to hold the meeting virtually, and everybody pulled together and made that happen. But I am so happy that this year we are actually here in person, and to get everybody energized, I of course wore my rhinoceros socks today just to get everybody in the spirit of our wonderful meeting. Now, everybody knows that the Cleveland Marathon is here. In Cleveland, we run a marathon actually every week to stay in shape, just to let you know, even during the winter. Unfortunately, Vikram Krishnamurthy, who is our local arrangement chair, is stuck over the next bridge and is unable to get here. So I'm going to butt in and give you his welcome as well. He wanted to welcome you to our wonderful city of Cleveland. We hope you enjoy the meeting and our wonderful outdoor activities. Of special note tonight will be the President reception held at one of my favorite places in town, the Rock and Roll Hall of Fame. And during that, I will be happy to give anybody a tour. I will show you all of the trivia that I know and love there, including John Lennon's green card. And one of the other kind of interesting things that you'll see there, you'll realize that Jerry Garcia from the Grateful Dead has now become an artist, and he now has a whole exhibit of his artwork there, and here's one of his Jerry Garcia ties. So I just want to welcome everybody to our wonderful city. Tom has a great program in store, should be very exciting, and wonderful to see everybody in person tonight.
As the next order of business, I'd like to call Dr. Janice Pasieka up to the stage. I presume Janice is somewhere in the room. As you know, last year virtually, Janice was awarded the Oliver Cope Meritorious Achievement Award. This is an award that is not given every year. It is given to a member of the AAES in recognition for contributions in the field of endocrine surgery as an investigator, teacher, and clinician surgeon. I think we can all agree that Janice did not fulfill just one of those categories, but in fact all three. So We actually struggled quite a bit as to whether to award it last year in virtually and not wait until this year, but I think in this was the best option, was to award it last year and then still be able to give Janice a hug this year as well. So Janice, thank you.
I'm supposed to say something.
I've never seen you at a loss for words, Janice.
You saw me at a loss for words last year when this honor was bestowed upon me, and I'll be honest that I reflect on that moment all the time. I'm still very chuffed about it. I'm still very humbled by the honor, particularly because it came from the membership. And when I have a bad day, I reflect upon that day when you surprised me and gave me this award. And I just hope I can live up to the standard of the Oliver Cope recipients and influence anybody here in the audience the best way I know how. And thank you.
So as you all know, yesterday there were 2 courses, the second AAS RFA course, faculty noted there on the right side of the screen, and the ultrasound course, faculty noted there on the left side of the screen. I'd like to thank the faculty for their efforts and their endeavors in these courses. Really outstanding from everything I've heard. Both courses were outstanding. They were both sold out actually quite quickly. And so we do intend to run additional courses. Hopefully the next one will be sometime in the fall. But these are, I think, are very important for keeping our membership and us at the cutting edge of technology. So thank you to the course faculty and course directors for superb efforts. Self-nominations. So as you know, a year or 2 ago, we initiated the process of self-nominations for committees. The AES has a large number of committees now, which do actually a tremendous amount of work. The committee members that are probably many of you in the room recognize the tasks that need to be done to keep this organization going. And I think we are very blessed to have such an enthusiastic membership. We never have trouble filling committees, but if you are interested, there's always more to do, and pretty much every year we seem to be adding a committee or 2. So please, if you want, scan that code. That's how you can complete that will take you right to the self-nomination form. And if you complete that by June 10th, that would be, you'll be eligible to then be on a committee. So please avail yourself of that opportunity if you're not already serving on a committee and have interest. I'd like to now call Dr. Ali Abadi, the chair of our membership committee, up to the stage to introduce and welcome our new members.
Good morning. It is my pleasure to introduce to you on behalf of the membership committee our new active and corresponding members. They are a particularly accomplished and personable group of individuals, and I hope over the next couple of days you'll find the opportunity to seek them out and congratulate them personally. If I could please ask the new members when I call your name to come up and line up in front of the stage so that we can recognize you as a group, and if you could please hold your applause till the end. Dr. Ayas Al-Khalili, Dr. Stephen Bruch, Dr. Rebecca Campbell, Dr. Jennifer Cannon, who's joining us virtually, Dr. Jason Cohen, Dr. Nita Erengeri, Dr. Lindsay Kuo, Dr. Brendan Finnerty, Dr. Maria Bates, Dr. Priya Didia, Dr. Mahsa Javid, Dr. Melissa Mao, Dr. John McAuliffe, Dr. Alexandria McDowell, Dr. Pavel Nokal, Dr. Sirinia Parservit, Dr. Jessica Schenk, Dr. Hadiza Kazar, who's joining us virtually, Dr. Chichi Stuckey, Dr. Lata Pasupuleti, Dr. Ludovico Senham Jr., and as far as our corresponding members, Dr. Taeyeon Sung, who's joining us virtually, and Dr. Frederick Trippinet. Please join me in welcoming them. Congratulations, and we hope to see all of you at the new member breakfast tomorrow. You may return to your seats.
Thank you and welcome to our new members. So now I'd like to call up Dr. Ben James, who will introduce the 2020 LaGerfo and FICA Award winners, who will then give a short presentation each on how they've, what they've done with their award. Ben.
Good morning. Just to put in a quick plug, so for anybody interested in applying for any of these awards, please don't hesitate to reach out to anybody on the committee. We encourage anybody interested at all, whether it's basic science, health services research, education, happy to talk to anybody at any point. For the first award presentation for the Paul LaGerfo Award winner, we have Samira Sadowski, presenting this virtually on novel treatments targeting epigenetic regulation of somatostatin receptor type 2 expression in radionuclide refractory neuroendocrine tumors.
I believe this is a virtual presentation, so do we have that ready to run?
An endocrine surgeon scientist at the NCI. I thank the AES committee for having selected my work and giving me the opportunity to present today my work on novel treatments targeting the epigenetic regulation of SSTR2 expression in radionuclide refractory neuroendocrine tumors. I have no disclosures. I'll present a brief background, my research aim, some of our preliminary results, as well as ongoing and future plans. NETs are rare, with an increasing incidence of 7 per 100,000 in the US, particularly increasing for the pancreas and GI tract. The main line of treatment to render patients disease-free is surgery. NETs are unique in that they express somatostatin receptor type 2. No mutation has been found in its gene, and its expression is thought to be epigenetically regulated. Our group and others have found its expression to correlate inversely with grade, and the loss of SSTR2 is an independent prognostic factor of worse prognosis. SSTR2 can be targeted for diagnosis and therapy. Our work at the NIH contributed to the FDA approval of 68 gallium dotatate PET-CT, which we found to be the best staging modality. 177 lutetium dotatate can be used as therapy in well-differentiated metastatic NET. The pre-requirement for this therapy is that patients have SSTR2 positive status. This has shown a progression-free survival benefit of lutetium versus control confirmed in the follow-up study. We are limited in terms of therapy for metastatic and high-grade NET, in part because with increasing grade, SSTR2 expression is lost. In the lower grade tumors, we can give octreotide and they respond to PRT. For pancreatic patient cases, we can treat with everolimus, sunitinib, or Captan. In high-grade G3 tumors, we are limited to platinum-based chemotherapy and streptozotocin. As many of these therapies are dependent on SSTR2 expression, our thought was by increasing SSTR2, we could improve treatment options for high-grade neuroendocrine tumors. My goal is to develop novel therapies for radionuclide refractory neuroendocrine tumors. I plan to achieve this by increasing SSTR2 expression and understanding its mechanism of regulation. I'll help myself by developing tumor models as well as translating my results into clinical trials. And beyond SSTR2, I'm interested in identifying new genetic and epigenetic drivers of tumor progression. We performed DNA methylation arrays in drug-treated cells and found promoter methylation to inversely correlate with SSTR2 expression, as shown in this figure. For example, QGP1 here has a very low SSTR2 expression. We validated this in publicly available PNET human samples, as well as in our NIH cohort of 94 patients, where grade correlated with methylation status. In vitro, by using HDAC inhibitory therapies, we managed to find a dose and time-dependent increase in SSTR2 surface expression. By using a reliable xenograft model and treating for 10 days by IP with HDAC inhibitor tacitidine and then collecting these tumors, we found a significant increase of cell surface SSTR2 expression. For clinical relevance, we then injected lutetium dotatate and found a significant increase of radioactivity within the xenograft tumors at the higher dose of tacitidine therapy. Using that same model, but injecting a therapeutic dose of lutetium dotatate, we showed a significant tumor volume reduction in the sequential combined therapy of HDAC with lutetium in pink compared to lutetium alone in green. And you can see here the representative tumor volume sizes. For our ongoing projects, our proposed epigenetic silencing mechanism for the SSTR2 gene promoter by closing the chromatin, we found increased DNMT3B de novo methylation to be a source of silencing SSTR2, as well as the loss of activating H3K4 methylation and acetylation, and the gain of repressive PRC1 and 2 histone marks. We validate this, for example, by using selective inhibitor ORY1001, which is a LSD1 inhibitor, and a H3K4 then demethylases, and results in an increase in SSTR2 expression, as you can see, at nanomolar doses. We further, by using single-cell ATAC-seq, looked for possible SSTR2 enhancer. On this UMAP, you can see that the cell lines 7 and 8 cluster away from healthy islet cells in yellow and green, and from acinar and ductal cells. And by looking at the peaks of open chromatin, we find possible enhancers that are then lost in our cell lines. We are currently validating this data in human PNET samples. impact do I intend to make in the field of neuroendocrine tumors? By upregulating and understanding the regulation of SSTR2, and then applying this with a clinical trial, I intend to improve targeted therapy in otherwise non-responding refractory patients. By finding information on epigenetic landscapes and the cell of origin, by using a tumor progression model, this will allow us to select neuroendocrine tumors according to clinically actionable target and stratify on prognostic outcomes. By developing a tumor model that is patient-derived and more representative of the tumor biology, this will allow for better studies in the field, as well as through creating a biobank, will generate data that inform us on the molecular profiles. Lastly, I want to thank my team, Dr. Jim Madigan and Rupali Sharma, and all the students that came through my lab, as well as in particular my mentors, Marta Zeiger, Pete Cherokee, and Carol Thiel, and all my collaborators. And I thank you for your attention, and I'm happy to answer any questions.
Great. Our next presenter that also won the 2020 Paul Lajerfo Award is Dawn Effelbaum from University of Wisconsin, and she'll be presenting on randomized controlled trial of preoperative steroids in autoimmune thyroid disease.
All right, good morning. Thank you for the privilege of the podium this morning. Thank you to the committee for this award to allow me to do this work. And I want to take the first 2 minutes of this talk actually to talk to you about a different side of Paul Lajerfo. I think sometimes Paul Lajerfo, there's some people in this room that remember him and knew him. There's other people in this room that don't know him very well. And Paul Lajerfo was a thistle sailor. And this is Paul Lajerfo sailing his boat. A thistle is a high-performance, one-design, 17-foot racing dinghy. One-design racing means that everyone races the identical boat, so the skill of the sailor is rewarded. It's kind of like the NASCAR of sailing. The first thistle was built back in 1945, and there's lots of thistles in circulation right now. 12 years ago when I met my now husband, he had a thistle, 3517 was his boat. And so over the past 10 years, I've spent hundreds of hours on this boat, hiking out on the rail. It is not a comfortable boat. It's a difficult boat to sail, and it really is for racing. It's sort of a gritty boat. We actually have fun doing this, as you can see us through the window here smiling at each other, which a lot of married couples who sail together don't often look like this on the boat. And we actually were just at Thistle Nationals yesterday morning, so 10:30 a.m. yesterday. We left Jacksonville, Florida, and drove up here trailing the boat to get here in time. We finished 26 out of 49, by the way, but we finished second in the last race, which was very exciting. Anyway, in 1977, which is the year that I was born, Paul Lajerfo was developing the thyroglobulin assay. He must have been working too late in the lab one night because he was looking for something else to do. And he wandered onto the grounds of the Nyack Boat Club, which was down the street from his house. This is Paul Lajerfo in the middle there, around maybe the 80s, early 80s or so. Boat clubs are these communities. They're not places where you just go and sip cocktails on a boat, on a yacht, right? They're gritty places. You got to do the work. So Paul would mow the grass and cook food for a bunch of sailors. And people at the Nyack Boat Club who still remember Paul and who talked to me about him last week, most of them said they didn't even know he was a surgeon for several years. He was just another guy at the club. He bought a Thistle. He bought Thistle 616. It was a beautiful old wooden boat, and he learned how to sail it. He was not a great sailor. He was very impatient with himself, people would tell me. But he would go out all the time, and he would sail it, and he got better. And he just loved that boat, and he spent a lot of time on it. And he taught other people to sail. The thing that struck me most about Paul is that in the 80s, he bought a new boat for himself. He sailed it for a while. He got some young people to sail it with him. And then after a couple of years, he gave the boat to the person that sailed with him most. A thistle this today would cost about $35,000, so it's like buying a new car and then giving it away a couple years later. He bought a second boat, he did the same thing, he gave it away. So in that way, he grew the club. He was a real ambassador for thistle sailing at Nyack, and that's what people remember him for. So I wanted to let you all know that this is one of his boats, 3750 or 21, which is still in circulation today. This was just at Nationals last week, that boat was sailing. So now on to my work. Graves' and Hashimoto's disease are autoimmune conditions increasingly being treated with surgery. High circulating autoantibodies of either of these diseases is associated with worse quality of life, even in patients who are euthyroid. By now, everyone in this room has probably read this study, a Scandinavian study randomizing patients who are euthyroid Hashimoto's to having surgery or no surgery. And all those little graphs below show that the quality of life in patients who had a thyroidectomy for this disease go way up 6 months after surgery and continue to trend up almost towards the baseline of the population. So since this study has come out, I think we're going to start seeing more patients with Hashimoto's coming to us for surgery. We know that rates of recurrent nerve palsy and hypothyroidism are higher in both Graves' and Hashimoto's patients. And most of the studies out there you see are comparing rates of thyroidectomy complications in those patients to patients who don't have inflammatory thyroid diseases. To me, I think that's sort of like comparing apples to oranges, right? So that's only risk, and complications is only one side of the equation about whether a patient has surgery or not. If we're starting to see more data that shows that there clearly is a lot of benefit in these patients, are some patients willing to accept that higher risk? And so my question really is not, can we compare more apples to oranges, but can we just shrink the size of this apple, which is the risks of thyroidectomy due to inflammatory conditions? So based on our anecdotal experience in a few patients that we reluctantly operated on who had hugely inflamed glands and very high levels of TPO antibodies, we gave a few of those patients steroids, and it seemed to make surgery at least a little easier than we thought it would be. So we hypothesized that giving patients a small dose of steroids before surgery might make a difficult thyroid less difficult. Furthermore, we wanted to take a look at some antibody levels and drew lots of different antibody levels to see what happened over time. So to test this, we designed a small pilot randomized trial of patients with either Hashimoto's or Graves'. Patients got 20 milligrams of prednisone a day in addition to usual preoperative treatment, which for Graves' patients is SSKI and Tums. For Hashimoto's patients, we don't have a good regimen before surgery. Surgeons were asked to fill out our previously validated thyroid difficulty scale, and we tested antibodies and sent quality of life surveys, which I will be talking about in my paper, but not today. Our demographics are seen here, pretty typical for this type of disease, mostly female patients. Our Graves' and Hashimoto's were split equally. The Graves, or the steroid group, was slightly younger and slightly, what was the other one that they were? Slightly male and slightly more younger in that group. Our results, disappointingly, saw no statistically significant differences between the groups that got steroids and did not get steroids. But again, this is only 30 patients, so we didn't expect to see huge p-values. PTHs drawn in the PACU for patients who did receive a week of steroids before surgery seemed to be a little higher. So perhaps we had slightly better ability to preserve those parathyroid glands, but really not a huge amount of difference here. But I'm going to drill down into some things. Steroid side effects, there were really very few. A lot of patients said they did have some side effect, but most of them said that they were not so bothersome. They were transient and mild. I'm going to skip quickly through the slide because this is antibody levels that I'm still drilling into. But this is showing that vertical line in the middle is a line of 1, and that's a ratio of antibodies before and after surgery. The horizontal line is patients above that line got steroids and patients below the line didn't. The big picture that you're looking at here, the graph on the left, is showing the week before surgery and then the day of surgery. Some patients who didn't get steroid, actually their antibodies increased during that week. I can't quite explain it, but antibody levels were roughly the same. And everyone's antibody levels by about 6 weeks really had dropped, and there wasn't a huge difference. So bear with me for a moment on these Hashimoto's patients. I'm going to venture into some low N numbers here, but the purpose of the study really was to generate any pilot data that would justify a larger trial. And if we look just at the Hashimoto's patients, in the week before surgery, all of them had some drop in their antibody level, a 20% drop overall of antibody levels, of TPO antibody levels. When you look just at the thyroid difficulty scale, all the surgeons actually thought the surgery was easier in the patients who had gotten prednisone on almost all levels. So the friability of the gland, the size of the gland seemed smaller, the vascularity was less. And then we had some things that the surgeons could check. Was it extremely difficult to dissect the recurrent laryngeal nerve? In the steroid group, none of my partners checked that box, but in the no steroid group for Hashimoto's, a couple of them did out of 7, right? 2 out of 7. So these numbers to me are compelling that there's something going on here with the Hashimoto's. I think that the reason is because we already prep Graves patients really well. We don't have good prep for a steroid or for a Hashimoto's. So in conclusion, a short course of prednisone is well tolerated. Steroids may make surgery for Hashimoto's patients easier. We have more work to do here, and I look forward to this week talking to anyone who is interested in doing a much larger trial of this, because I think we need more numbers, and I think we're going to be seeing more of these patients. Thanks to my partners and Sarah Schaefer, who's our nurse practitioner, who helped with this. And one last picture of Paula Jerfwin's sailing legacy. There's an award given at his club every year to the person who is most generous to One Design Sailing. So his legacy lives on in the sailing world and in this room. Thank you very much.
All right. Next, I'd like to introduce our 2020 THICA Award winner, Taylor Brown from Washington University, who will be presenting on mapping of intratumoral heterogeneity and identification of druggable targets in anaplastic thyroid cancer using whole exome sequencing.
Thanks. Sorry for the long title. Let's see. You guys able to load the slides? Okay, great. Okay, so my name's Taylor Brown. I'm an endocrine surgeon at WashU. I have no disclosures. So the background, as many of you all know, ATCs are highly aggressive. The 2-year survival rate is generally reported to be about 20%. The molecular therapies, encouragingly, are showing promising results, and multiple regimens have now been recommended in society treatment guidelines. However, despite these interventions, patients frequently and ultimately, succumb to the disease. The likely cause of this recurrence is, or treatment resistance is caused likely by tumor heterogeneity, which is, not accounted for by the current clinical and research practice of single sample sequencing analysis. So in this model here of tumor evolution, we see a normal collection of blue cells. We then have a founding clone of green cells, and then through evolutionary forces, tumor heterogeneity develops throughout the cancer cohort, as signified by the different colored dots. Despite 1,400 cases of ATCs being sequenced and published, I think less than 1% of those cases have actually looked at this question. So with that in mind, our aim and hypothesis was to perform multi-regional next-generation sequencing of anaplastic thyroid cancers, and we were hoping that this would help us characterize tumor heterogeneity and clonal architecture, identify clinically relevant subclones not ordinarily identified by single sample sequencing, demonstrate unique regional specific druggable targets and or markers of treatment resistance. And we think, ultimately this would help inform decision making for molecular therapy design and treatment strategies. So far we've retrieved 6 cases from Barnes-Jewish Hospital after IRB approval. And then for each case we've performed NGS analysis anywhere from 4 to 8 different regions of the tumor. We've tried to pick regions that have variable histological growth patterns. We've picked regions with metastatic lymph nodes, and then as well as normal matched tissue. And then we performed whole genome sequencing at a depth of 60x. So this is just kind of an overview of the strategy we took. The black boxes here represent the ATC component. The green box represents normal tissue, and the blue box is the well-differentiated component if it's present. So I'll go through some of our results. I apologize, these slides are very busy, but I just wanted to give you some of the preliminary results we have so far. So in this first case, this is ATC3. We sequenced 8 different regions in the tumor. And I want to direct your attention to the bottom bar graph of the orange and maroon boxes. And what this signifies is that there's significant genetic variation between different regions of the tumor. So the orange boxes represent unique mutations only identified in that region, and the maroon boxes identify regions that are present throughout the cancer. And this is looking at all the exon mutations or the coding regions. And what you can see is that there's significant genetic variation from region to region. About 80% of the mutations we detect are unique to that region. Similarly, here we have an oncoprint of the whole genome, and just for the sake of not making it too complicated, we filtered out the silent mutations. And again, we see really a significant degree of genetic variation from one region of the tumor to the next. And then finally, if we kind of hone down on the actual region that we sequenced, this is a variance allele frequency plot, and I won't walk you through all the details of it, but what we even see just in sequencing one area, we can detect multiple clones in one area. So that's signified by the kind of the little two humps you see there with the arrows pointing at it. In another case, similar to the previous case, we see a lot of genetic variation, though perhaps not as much as the previous case. And then when we kind of pull out the mutations that are associated with thyroid cancer, have been proven to be associated with thyroid cancer, we see that there's significant variation in the repertoire of mutations we detect, including P53, cyclin-dependent kinase, retinoblastoma. These are all genes that are major drivers in cancers. And what this data suggests is that the different regions, based on these different mutation profiles, probably behave differently in response to molecular therapies. I'll just show briefly two more cases. I think the again we see a significant amount of genetic variation in different regions. Again, we're detecting tumor subclones even within the single spots that we're sequencing, and this is the final case as well. So that's where we've gotten so far. So I think we conclude that there's a significant amount of tumor heterogeneity in ATC. This heterogeneity includes variable mutations in thyroid cancer-associated genes. We think that in turn would cause the different regions of the tumors to respond differently to molecular and systemic therapies. And as such, I think this would suggest that we need to consider more comprehensive analysis of tumor heterogeneity when we make treatment decisions for these patients. Future directions, we have more cases we'd like to sequence. We have a lot of data to go through. There's 3 billion base pairs for each region we sequence at a depth of 60. There's a huge ton of data to churn through. But we need to also look at gene copy number alterations, the presence or absence of gene fusions, and then finally put together what we'd call like an evolutionary tree where we can kind of get a bird's eye view of how the tumor evolved from its very beginning to its before it was resected. And then ultimately we think this genetic variation will affect druggability throughout the cancer. I just want to thank my research team at WashU, particularly Ha Deng there. He works at the Genome Center. I'd like to thank my clinical colleagues, TK Pandey and Will Glanders, obviously the AES, and THICA for supporting for me with this grant, and happy to take any questions if time permits.
Great, those were excellent presentations. I would just mention that the 2022 winners will be announced during the awards session on Tuesday, so look forward to seeing you all there.
Thank you, Ben. Those were 3 great presentations, and I think a testimony to the need to continue for the AES to continue to fund these lines of research. Because it really was fabulous. So we're actually running right on time, which is great. The next we have the UCSF Carol and Orlo Clark Distinguished Lecture.